Okay. Yeah, thank you very much for having me today. The title of my talk is Unifying Traditional Knowledge with Phylogenies to Enhance the Usage of Biodiversity Resources. And it's part of a work that I did back at the University of Reading in collaboration with Kew Gardens and Imperial College London. So in today's talk, I'll be trying to convince you that combining data from traditional knowledge, which is a very important part of culture, with biological phylogenies actually makes sense. And to do that, I'll be explaining to you what we did in a study that was published recently, which uses data from traditional medicine, places that in a phylogenetic framework, and tries to draw on that in order to inform bioprospecting. So I'm talking about traditional medicine, and in particular herbal traditional medicine. And for those of you who don't know much about it, although to some extent we've all been exposed to it, but 60 to 80% of the world's population depend on traditional medicine for healthcare. There's 10 to about 50,000 plant species used traditionally for medicine in different parts of the world. And obviously that body of knowledge has contributed to the development of pharmaceutical drugs as well. So about a quarter of drugs that we find in pharmacies nowadays are plant-derived, and a lot of those in the initial days of pharmaceutical drugs came from plants that were traditionally used in different cultures. Now there's obviously a lot of interest in what's called ethnobotany, which is the study of traditional use of plants. And there's, in the last decades, there's been huge efforts in compiling a comprehensive list of plants used in different parts of the world, both to preserve traditional knowledge, understand how traditional medicine might work, and also direct the discovery of new medicinal plants and natural products, which is what I'll be focusing on today. So there's a vast diversity of plants in the world. There's something between 250,000 to 400,000 species, but only very few of those have been screened for bioactivity. And there's been no systematic study to determine whether the plants used in traditional medicine are actually underlined by bioactivity. So there's a lot of skepticism around traditional medicine and whether it works and whether it doesn't. And a lot of that has had to do with the fact that methods for selecting the plants to be tested for bioactivity at the first place were a bit faulty. So there were no consistent methods, largely speaking. And a lot of people have just gone out there and tested plants they just found in nature, or obviously plants that came from traditional medicine. But one criterion for selecting a plant to then put it into the lab and test for bioactivity is what's called the independent discovery by different cultures. And what that basically tells you is that if you've got a plant species that is used by different cultures, and if that usage stems from independent discovery, then it's much more likely that this plant species is actually bioactive. And yeah, the idea behind it is very simple. If different people came to the same conclusion, then maybe these people are right, whatever right means in this case. So ethnobotanists have tried to do this sort of cross-cultural comparisons in plants that people use in different parts of the world. But in order to be confident that the discovery is independent, you have to select cultures that are very disparate. These cultures are usually found in very different parts of the world. There's little floristic overlap between the different parts of the world where different cultures are exposed to. And it's been very hard to draw conclusions of common patterns in traditional medicine in different parts of the world. Now, what we did in this study is we brought molecular phylogenies into the mix. And molecular phylogenies is useful in this case because instead of actually trying to find species that are used in common, you can calculate the evolutionary distance of any species used in a part of the world to any other species used in another part of the world. And therefore, calculate some sort of relatedness of medicinal floras in different parts of the world. And here's what I mean. Here you've got two different medicinal systems, one in green and one in orange. And you've got species that are found in different places. So the dashed lines are the species that culture green is exposed to, and the solid lines are the species that the culture orange is exposed to. In both cases, these cultures use no species in common. But in the scenario to your left, these cultures have used the closest relative in every single case. So although they don't use the same plants, they've done the closest thing to using the same plants. Does that make sense? Good. So, yeah, for this study, I focused on three different cultures. So, groups of cultures, I should say. I used the cultures from the Cape of South Africa, Nepal, and New Zealand. And I use these places because they're very far apart, and people there are exposed to very, very different plants. So the floras are dramatically different. And I built a phylogenetic tree for each of the floras. I used one species per genus. I sequenced a standard plastid marker, and yeah, I reached a sampling of between 80 to 90% for each of the flora, and built a tree with maximum likelihood. And I also compiled a lot of ethnobotanical information from the literature, so lists of all species that people have recorded that people use in any of the three areas. And also broke that down to 13 categories of use, so depending on what the species is used for. So things like cardiovascular and musculoskeletal, and blood purity, and so on and so forth. And all that information was then mapped on the phylogenetic trees. So the first thing that we found was that those uses were phylogenetically clustered. There was a very significant pattern in what species people use in traditional medicine. And that's something that ethnobotanists have known for decades. Not all plant groups are included in traditional medicine. Some are more likely to be included or not, but this is the first time that it was shown at using explicit phylogenetic tools. However, the question was, I'm going back to the independent discovery in different cultures. So, okay, people use specific lineages, and not all lineages in traditional medicine, but do they use the same lineages in different places in the world? So to do that, I combined all the data, the sequence data, and essentially I built a big phylogenetic tree that represented all three floras. So it's kind of like taking the plant tree of life that these three floras are exposed to. And what I found is that the distances on the phylogeny between those medicinal floras were significantly smaller than expected by chance. So what seems to have happened is the scenario that I showed you earlier. So people have used very, very similar, I should say closely related species in traditional medicine. And not only was that true for the 3 medicinal floras in total, but when I looked at the categories of use, that was also the case. So people used very closely related plants for cardiovascular problems and for musculoskeletal problems, and so on and so forth. So yeah, these distances on the phylogenetic tree were significantly smaller than expected. So I probably haven't explained very well what phylogenetic distance is, although it might be straightforward for some people, maybe not. It took me some time to get my head around it. So yeah, phylogenetic distance is the distance from any given species to another species on the phylogenetic tree. So these 2 species have a very small distance because they're closely related, whereas this and one of the species on the top are very distantly related, so the distance will be smaller. So yeah, when we say that species use, that people use species from that are closely related, yeah, it's the scenario you see there. So there are many explanations that why that pattern is there, and ethnobotanists have discussed that again in previous days before phylogenies were even around. So yeah, it could be cognitive patterns underlying people's selection of medicinal plants. So things like morphological characters that are very important for inclusion of species in ethnomedicine and are also constrained on the phylogeny could drive this pattern. Or it could be chemistry and therefore bioactivity underlying these patterns. Again, we know that in many cases chemistry is a character that's conserved on phylogenies. Closely related species are likely to share chemical profiles. Either way, yeah, I think the inclusion of closely related plants in ethnomedicine in different cultures is a product of relatives sharing other morphological characters or chemical properties. But the main direction with this was to feed into bioprospecting, so the discovery of new natural products. And the way to do it was to identify what we call the hot nodes. So these are parts of the phylogenetic tree that are used heavily in ethnomedicine. So when I showed the 3 trees earlier, those red branches that are really heavily used in medicine. And I won't go into detail about how we did that, but you can ask me later or read the paper, or both. And what we found that these hot nodes could actually inform bioprospecting scientists. So a lot of pharmaceutical companies nowadays that look for natural products do what we call a random selection of species. So they just go around nature and choose species A, B, C, and D. So if they were to do that, the coverage they get for the floras, in this case, would be the blue bar that you see there. And I've got the results for the medicinal floras as a whole and the categories of conditions, so the more specific usages. So the hot nodes that I identified on those trees include about 30% of the flora. So if someone was to go around nature and pick up 30% of the flora, we'd expect, if collecting was random, that they'd also end up picking up 30% of the medicinal flora. Which was not the case. So if you actually select the plants that the hot nodes tell you to, you get more of the medicinal plants. so you get an increase of species that, of medicinal species that you could get. And I'm running out of time, so I'm not going to get into detail. But essentially, what this tells you is if you select, if you go out and select species that are found in those hot nodes identified by phylogeny, you're more likely to find medicinal species in nature. And we wanted to see, there was this really cool study recently that showed that organisms that have produced pharmaceutical drugs are also clustered in the phylogeny. And although what you see here is not a real phylogenetic tree, it's like a taxonomical phylogenetic tree, I still think it's very cool. It shows a very similar pattern that plants that have produced drugs are from certain lineages and not all lineages. So the obvious thing to do was to compare those lineages with the lineages we identified based on traditional medicine. And what we found is that, again, if you look within those hot nodes, you've got significantly more of those species that produce drugs than expected by chance. So, yeah, all in all, I think what we've shown here is that phylogenetic relationships of species underlie pupil selection of medicinal plants in traditional medicine. We've got closely related plant species used in different parts of the world, and these groups coincide with the ones that produce drugs. We find potentially independent discovery of efficacy and also large-scale evidence that plant bioactivity underlies traditional medicine. And similar patterns have been shown for other taxonomic levels and other properties as well, such as edible plants. And a few colleagues of mine and myself call this phyloprediction, so using phylogenies to predict what species might be useful. And it can be, what we're working on and other people can try as well, is do similar analysis for more areas and using more detailed phylogenies, different organismal groups and different target properties. And I think especially as sequence data become more and more available, I think it's going to become easier to generate large phylogenetic trees. So yeah, thank you very much.